Module 2, Molecular Biology Techniques and Applications. Lecture 1, Introduction to Molecular Biology Techniques. Molecular biology focuses on the structure, function, and manipulation of nucleic acids and proteins. The techniques in this field allow researchers to explore genetic material and design experiments to sense, control, or modify biological systems. This module will cover protein purification, enzyme assays, molecular cloning, PCR, protein crystallography, and enzyme kinetics, among others. Lecture 2, Protein Purification Methods. Proteins must often be purified before their properties can be studied. Steps include homogenization, breaking cells open to release proteins. Differential centrifugation, separating organelles from soluble proteins. Gel filtration chromatography, separates proteins by size. Affinity chromatography, isolates proteins based on binding specificity. Purification of enzymes such as lactate dehydrogenase, LDH, is a classic example. Lecture 3, Enzyme Assays and Protein Assays. After purification, proteins must be quantified and their activity measured. Enzyme assays measure catalytic activity under controlled conditions. Protein assays, methods such as Bradford and Lowry assays measure protein concentration. Characterization of LDH includes determining kinetic properties, inhibition, and substrate specificity. These assays allow researchers to evaluate protein function. Lecture 4, Western blotting and protein characterization. Western blotting, introduced in Module 1, is also key for protein characterization. By using antibodies, researchers confirm the identity, size, and abundance of proteins in a sample. Combined with enzyme assays and chromatography, Western blotting helps construct a detailed picture of protein properties. Lecture 5, Protein Crystallography. Protein crystallography is one of the most powerful tools for understanding protein three-dimensional structure. Steps include crystallization, X-ray diffraction, and structure determination. Knowing a protein's structure reveals how it works, how it binds ligands, and how drugs might target it. Lecture 6, Polymerase Chain Reaction, PCR. PCR is one of the most transformative molecular biology techniques. It amplifies specific DNA sequences, making millions of copies in a short time. Key steps include denaturation, annealing, and extension, repeated in cycles. PCR enables cloning, sequencing, and diagnostics. Lecture 7, Ligation, Transformation, and Screening. Molecular cloning often involves inserting DNA fragments into vectors. Ligation, joining DNA fragments to plasmids using ligase enzymes. Transformation, introducing recombinant DNA into bacteria. Selection and screening, identifying bacteria that carry the desired construct, often using antibiotic resistance or colorimetric assays. These techniques allow scientists to design and manipulate genetic material. Lecture 8, Enzyme Kinetics. Enzyme kinetics is the study of reaction rates and how enzymes interact with substrates. Parameters such as Vmax and Km describe catalytic efficiency and affinity. By studying kinetics, researchers understand enzyme mechanisms, inhibition, and regulation. Applications include drug development, diagnostics, and metabolic engineering. Lecture 9, Applications of Molecular Biology Techniques. The techniques we have studied are applied in many areas. Medical research, understanding diseases, developing vaccines, and diagnostics. Biotechnology, production of recombinant proteins, enzymes, and hormones. Agriculture, genetic engineering of crops for better yield and resistance. Environmental studies, detecting pollutants or engineering microbes for bioremediation. Module 2 Summary. In this module, we covered essential molecular biology techniques, protein purification, enzyme and protein assays, Western blotting, protein crystallography, PCR, cloning methods, and enzyme kinetics. We also discussed their applications in medicine, biotechnology, and agriculture. Course conclusion. This course on techniques in biochemistry and molecular biology has introduced you to the major laboratory methods used to study biomolecules. Module 1 focused on the principles of instrumentation, electrophoresis, chromatography, spectroscopy, centrifugation, microscopy, and related techniques. Module 2 explored molecular biology methods, including protein purification, enzyme assays, Western blotting, PCR, cloning, and enzyme kinetics. Together, these techniques provide the foundation of experimental biochemistry and molecular biology, equipping you with the knowledge to design, analyze, and interpret experiments that reveal the molecular basis of life.